the old way we did things can be costly and painful.
Current detection technologies, however, are very limited. Everyone has the right to use these technologies.
It's really thrilling how we make use of the latest in material science and the whole world of nanotech.
This will not be possible without everyone's contribution.
Good afternoon, Dr. Shao. It's really a pleasure to have you back with us again. Today's topic is something that's close to everyone's heart, which is the COVID-19 situation globally.
Yes.
What is so important about testing? Because not everybody believes in testing. And the whole world is in this, but from a scientific point of view, why do you think it's so important?
Diagnostic or testing is very important across different diseases, but the COVID-19 situation, the global pandemic, sort of bring out a very, very special and strong need for testing at very large scale, particularly because we are trying to locate patients for contact tracing, for improving public health, and most importantly, actually to address personal needs, so that if a patient actually contacts the disease, it will be a lot easier, right, and to treat, to monitor, and also to improve better health outcome.
Where is the world at on this, and can you give us an overview of COVID-19 testing? Because I think everyone would love to hear a scientist's up-to-date point of view about this.
The COVID-19 situation actually has brought in a lot of technologies to focus onto this disease because of this global need for testing. We can generally divide them into two major categories. One of them is what we consider about testing of the virus, meaning this is the viral component. Another type of test is actually we are looking at our body's response to the virus. One very good example is actually the antibody test. This is actually looking at our body's immune response against this disease. Between these two tests, or two types of tests, the ones that we are actually testing for the viral materials, these have good examples such as the PCR testing for nucleic acids, as well as testing for some kind of these viral antigens. All of these materials belong to the virus. They originate from the virus. What essentially they're telling us is, are there active viruses in our body? How infectious are we at the point of testing? The tests that are looking at our body's immune response sort of indicate to us that we had recovered from an infection. So to me, these two tests actually serve very different purposes. If we are actually trying to control infections for public health perspective, then the tests that are looking for these viral components to determine whether the individuals are infectious will be very valuable for quarantining, for identifying these active patients. But the ones that are actually doing measuring our body's response, these will be useful for contact tracing purposes, so that in advance we'll know who are the close contacts of patients who have recovered from the disease and therefore might now be in a state of the active disease.
If you could explain to us how the current PCR tests are done and why there are so many, and why some are accurate and some are inaccurate or not so specific, and even false positives for everything in that arena, and what is happening there, and what are you doing that's really quite different? Maybe you could run us through?
Okay, yeah. So COVID-19 is actually caused by this virus that's known as the SARS-CoV-2. It's a very unique RNA virus. We'll go a little bit more into the detail about this virus and why and what are the ways that we are currently adapting technologies that we have already developed, let's say, for example, PCR for this detection, and what are some of the challenges in detecting this virus. So currently triaging for this virus is difficult because the symptoms that patients present, at least at the early time point, it's very common type of respiratory symptoms. But because the disease is so infectious and can cause deadly complications, it's very important that we detect and do early diagnostics so that we can reduce the spread of the disease. As we just talked about the different strategies at which we are detecting the viral components, or in particular the nucleic acid component of this virus, we're actually learning very much from nature. In all of our cells, the cells in our body, the cells are actually undergoing a replication process where we are actually copying genetic material from a parent cell and then making multiple of these daughter strands in the daughter cells. So it's a process that we're actually learning from nature. And scientists have used this knowledge of how cells are doing in nature to develop this technology called PCR technology, which is currently the gold standard for nucleic acid detection in COVID-19 diagnostics.
So originally it's gene detection based on DNA material that now we are applying to viruses where their genetic material is RNA.
Right, yes. So as we discussed, the COVID-19 virus is actually an RNA virus. But what we have learned and what we have developed in all of these technologies are actually a DNA way of detection to make multiple copies of DNA and then doing the detection. So for this PCR detection, in order to bridge that difference between what we have, which is SARS-CoV-2, which is an RNA virus, versus a DNA detection method, we actually have to process the samples in a couple of ways and then making the final detection. So the different steps in a gold standard PCR detection currently involves, first of all, we have this viral component that contains a RNA as the genetic material of this virus. So the first step of getting to this test is actually to do a extraction of this genetic component, which is the RNA. And in this step, very, very clean RNA will have to be obtained from the virus, because should we have any other contaminants in the sample preparation, then it will interfere with our downstream assay. And after we have successfully gotten out the clean RNA, then we have to convert it to DNA, which is another type of genetic material. But that is the one that PCR technology can work with. PCR technology by itself cannot directly work with the RNA molecules from the viruses. So after we have converted the material into a DNA, then we move on to change the DNA, the target DNA, which is the mother copy, into many, many, many copies of these daughter or progeny strands using a way that very much like a molecular photocopier, that we're actually making many copies of these daughter strands. And this is a step that we actually learn from biology of the cells. And in this step, we're actually putting the sample through multiple cycles of these temperature changes, from very high temperature to low temperature, high temperature, low temperature. So many copies of these cyclings of these reactions to get to these many copies of these daughter strands, and then we do detection. So some of the challenges are that firstly, there could be reagent shortages because every single step of this requires dedicated reagent. Secondly, there's a very long operation time. As I already described about these processes, they are not trivial. So typically they can take from 4 hours to 12 hours. With large-scale screening, hospitals are actually using very dedicated equipment, because all of these steps will have to be done in very, very clean setting, that we cannot have a lot of contaminants in the solution. And they will also wait for a lot of samples to come in and then make a measurement through a large device. So that all of this will add to the waiting time for results.
So why are they all so different?
In making these molecular reactions work every single test will have their secret recipes of material that they are putting into the test, such that they are identifying specific regions in the genetic component of the virus and then making multiple copies from that genetic component. Because of the multiple things and the secret ingredients that they're adding to the reaction mixture, they're all detecting different things. Different things will have different performance. And therefore, in terms of diagnostic accuracy, we are seeing quite a garden variety of performance in today's setting.
And I think the other problem with all this is that the minute we talk about mutations, we're finished, right? We got to start from ground zero again, right?
Yes, it's related with how we actually use PCR, or how PCR technologies are being designed and developed. They have a requirement that you need to find dedicated region and then make multiple copies from these regions. So should there be a mutation, then my ability to identify that region will have to be revisited and redesigned, so that we are now looking at new targets, new amplifications, and then to see whether evaluate the performance again.
I think, for the audience that maybe are not familiar, in every cell there are genetic material that replicate, and based on that, whether it's a virus or bacteria, that's how they multiply. And what the scientists have so elegantly done is to find regions of this to detect whether indeed the RNA or the genetic material of the virus is actually present in someone's cells. And to make it even worse, as many of you have heard, what are mutations? They're just changes in the genetic material of the COVID-19 RNA. So it's very, very hard for the world to catch up in general. I'd like to ask Dr. Shao to share with us this phenomenal new way they've designed, a molecular switch that can overcome all this, and maybe this is really a game changer that can be shared with the world at large.
To overcome a lot of these challenges of traditional and conventional PCR technology, we developed a new technology that's called the Envision technology. In this technology, we are actually building molecular switches from nucleic acids as well as proteins, couple them together such that now this becomes a molecular machinery that can help us to detect COVID-19 RNA directly in 30 minutes, entirely at room temperature. So bringing this type of detection, very exquisite and sophisticated RNA detection, to the patient level, almost in a doctor's office. We built this technology based on this concept of molecular switching. So we use genetic sequences as well as protein complexes to couple them together to form this molecular switch, which is a molecular machine. In the presence of the RNA target, which can come directly from the COVID-19 virus, this binding of this target will cause the switch to be activated because this molecular complex now dissociates. In the presence of the target, the molecular complex is turned from an off state to an on state, such that it liberates a very active enzyme. So in this case, we are actually doing an information conversion from an RNA target into an active enzyme. The active enzyme is a very potent molecular catalyst, which can then go on to activate and recruit other friends and catalysis reactions to happen, that in the end we will be able to generate a very strong signal enhancement directly from this clinical sample, which contains the RNA target. So a few advantages of this type of detection is that firstly, it allows us to cater to new mutations very quickly, because we are able to design this kind of switches without a lot of the design considerations that is causing PCR to be very difficult to be adapted for new mutations. The second thing is that with this type of molecular switching is an information conversion and a signal amplification process, such that we are able to achieve a very rapid and sensitive detection mechanism with minimal equipment requirement, that the entire technology can be bring closer to the patient level, away from a lot of these laboratory requirements, such as in a very extensive clinical lab. And also in terms of handling, you don't need a trained personnel to handle these type of processes.
Excellent. So you are not multiplying the number of daughter RNA, you're just amplifying the signal.
We transfer the information into an enzyme. The enzyme then go on to amplify the signal. We're not making any of these daughter copies in that way.
In this way, you're actually really overcoming almost all the problems, right? Because actually any trained practitioner of any sort, as long as you train them, they should be able to come out with the results within 30 minutes.
Yes, yeah. And that's why it's a completely different technology platform.
How far are you all at with this?
And we're currently in the progress of making this a commercial product. So with the hope of this Envision technology is that it's able to accelerate the diagnostic of this COVID-19, actually facilitate a lot of our daily lives, so that we are actually able to get results very instantaneously. And a lot of times, should this be possible, can also be done at the privacy of our home.
2 questions. When will this be ready? You anticipate that's commercially available?
We are hoping that we can actually bring this to market at the end of this year.
Have you started matching the data and
Yes, yeah.
versus even other test kits versus real life diagnosed patients? And
Yes. Yeah, so the technology has been clinically validated with COVID-19 patients as well as healthy controls.
Yes.
What we have found is that we are actually able to get a very good diagnostic accuracy of over 95%.
What do you expect the cost to be? Equivalent, higher, lower?
It should be significantly cheaper than a conventional PCR.
If Dr. Shao and her team can actually bring this quickly to the market, what will happen is we really don't need as much downtime in terms of even travel, because ideally this should be available at all the airports.
Yeah, so that's the hope, that we can actually make our contribution to the pandemic situation by really bringing all of these traveling daily activities back to normalcy.
Can you just tell us how many groups of institutes or people that got together to try to get this? I'm sure it's not just the NUS side, even inter-departments as well as clinicians, as well as global collaborations. Can you tell us how this came about?
This is a very, very good question. It's an interdisciplinary project where we have expertise in people who are developing these molecular switches, so very much on molecular biology. We also have folks from the device side, where we're actually building the microfluidics platform, such that from user perspective, it will be sample in and solution out. And finally, we are also working with clinicians who are helping us to validate this technology with clinical samples, and also providing very valuable feedback about what type of detection sensitivity we actually need to get to across different patients at what different time points of this type of detection.
Besides Singapore, do you all collaborate with other institutes overseas?
This is an entirely Singapore technology.
That's very, very nice to hear, because I can imagine you have to work very closely with clinicians to get this out.
Yes.
How long did this take the team?
So it has taken the team about half a year to basically finish adapting the technology for COVID-19, for virus detection directly from native clinical samples, and then also for the clinical validation.
Technically, to adapt, minus off the approvals, it's literally 3 months.
To adapt the technology for different clinical purposes is actually much, much shorter. We can finish in about 2 weeks. So some of the key challenges, right, for COVID-19 detection is primarily because that we want it to be a very point of care type of detection platform. So sample handling, liquid handling, these are components that we have to add to this molecular technology, such that it's a lot more user-friendly from a layperson perspective.
Thank you, Dr. Shah.
Yeah, thank you very much. Thank you.